Welcome back to this next video in the series of lectures on the cell cycle. In the last video, I've told you that the MCDK that was activated by the action of the CDC25, in the CDC25 that was actually removing the inhibitory phosphates from the active site of the CDK1, and the complex of the CDK1 and the cyclin B that was actually the MCDK that got activated. Now this MCDK that was performing some important functions. One of the function of this MCDK was the condensation of the genetic material, which it was achieving by the activation of this condensation complex. Another important function of the MCDK was the breakdown of the nuclear envelope, and it was achieved actually by the phosphorylation of the lemon protein, resulting in their repolymerization, and the phosphorylation of the nucleoporine polypeptides, thereby removing them from the nuclear envelope. Another important function was the assembly of the mitotic spindle, and the last important function that was actually the cessation of all gene transcription, because when the condensation complex they were going for the condensation of the genetic material, that actually result in the cessation of all gene transcription. Now in this video I'm going to tell you about the next phase of the M phase, that is known as the metaphase of the M phase. Now this figure is actually showing you the first phase of the M phase of the cell cycle, which is actually the prophase. Now I told you that the MCDK that was going for the polymerization of the microtubules, and the microtubule, they were actually polymerized by the conversion of the GDP of the beta tubulin into the GTP, and that was actually achieved by the GEF enzyme, and this GEF enzyme was actually activated by the MCDK. Now if you look at this particular figure, here you have gone for the polymerization of the microtubules. You have already duplicated the genetic material, but these microtubules, they will not be able to interact with the sister chromatid unless you get rid of this nuclear envelope. And this nuclear envelope that was actually dissolved by the action of the MCDK, we have already discussed that. Now if you look at the metaphase of the cell cycle, in the metaphase, actually the nuclear envelope that is gone now, so I'm not showing any nuclear envelope over here. At the in the metaphase of the cell cycle, you have got two clear poles, like this is one pole of the cell and this is the other pole of the cells, and you have got centrosome over here and you have got the centrosome over here. If you remember, I've told you that the sister chromatids, like this is a pair of the sister chromatids. So I'll be making the things that are attached to all of the sister chromatids, just giving you a single example over here. Like if you look at the sister chromatids, you have got the centromere over here, and you should differentiate between the centromere and the centrosome. Centrosome, they are the microtubule organizing centers. The centromere, they are actually sequences of the DNA. And to these centromeres, you have attached a protein complex that was the kinetochore protein complex. And we divided the kinetochore into the inner kinetochore and in the outer kinetochore. The inner kinetochore was in direct interaction with the centromere. The outer kinetochore was actually interacting with the microtubules that was originating from this centrosome. Now at this particular point, I want to focus on the different types of the microtubules and their point of interactions. Now if you look at this centromere, and if you talk about the centrosome, sorry, this is the centrosome and the other centrosome. Both of these centrosomes, they are actually responsible for a generation of three kinds of the microtubules. The first important microtubule that is known as the kinetochore microtubules, and as the name indicate, these are the microtubules. They are going to interact with the kinetochores that are already attached to the sister chromatids. So the kinetochore microtubule originating from this centrosome are going to interact with this member of the pair of the sister chromatids, and the kinetochore microtubule originating from this centrosome are going to interact with this sister chromatid of the pair of the sister chromatid, and this will be true for all of the pairs of the sister chromatids. So these kinetochore microtubules, they are in indirect interaction with the sister chromatids. By indirect, I mean that they are not directly interacting with the sister chromatids, but they are interacting through the kinetochore. So this kinetochore microtubule, by interacting with the kinetochore, are in indirect interaction with the sister chromatids. Another important microtubule that are originating from both of the centrosomes, they are known as the external microtubules. Now these external microtubules, they are not interacting with the sister chromatids, they are actually interacting with the cell membrane. So the external microtubule originating from this centrosome will be interacting with this part of the cell membrane. The external microtubule originating from this centrosome are going to interact with this part of the cell membrane. Another important microtubule that are originating from both of these centrosomes, they are known as the polar microtubules. And as you can see in this diagram, that these polar microtubules, they are not interacting with the sister chromatids just like the external microtubules, but these polar microtubules, they are actually interacting with each other. The polar microtubule originating from this centrosome, the polar microtubule originating from this centrosome, they are actually in interaction with each other. They are not interacting with the sister chromatids. So the only microtubules, they are interacting with the sister chromatids, they are the kinetochore microtubules. Once you have attached the kinetochore microtubules to all of the sister chromatids, you actually see a pattern like this, that all of these pairs of the sister chromatids, they are attached to the kinetochore microtubules from both of the centrosomes. By that I mean that if you have got this, if this is a single pair of the sister chromatids, so this member of the sister chromatid is interacting with the kinetochore microtubule from this centrosome, and this member of the pair is interacting with the kinetochore microtubule from this centrosome. So this will be true for all of the pairs of the sister chromatids. So if you talk about the human beings, after the S phase you have got 46 pairs of the sister chromatids, and all of these sister chromatids should be attached to the kinetochore microtubules from both of the centrosomes. We call this as the bipolar attachment. Now if you look at this figure, this is one pole, this is another pole of the cell, and this is one member of the pair of the sister chromatid. So it is attached to this centrosome. The other member of this pair is attached to this centrosome. So you are actually talking about the attachment of this pair to two poles, and therefore we call it as the bipolar attachment. This bipolar attachment is actually bringing all of the pairs of the sister chromatid to the middle of the cell, which we usually call as the equatorial plate. Now one important thing to keep in mind is that this bipolar attachment is very important if you want to move from the metaphase into the anaphase of the cell cycle, because if there is not bipolar attachment on even a single pair of the sister chromatids, the cell is not going to allow you to move from the metaphase into the anaphase. Now this bipolar attachment is actually ensured by an important checkpoint which is known as the spindle assembly checkpoint. And as the name indicates, this particular checkpoint is actually going to see about the assembly of the spindle, and that actually is responsible to check the bipolar attachment. So if you have got a single unattached kinetochore, there is no bipolar attachment on even a single pair of the sister chromatid, this spindle assembly checkpoint is going to halt the cell cycle at the metaphase, and you will not be allowed to move into the anaphase of the M phase of the cell cycle. We will continue this discussion in the next video.